We've all asked ourselves, what is DNA? Is it a simple molecule, a protein, or just a tiny ladder? Well, you're close. DNA actually stands for deoxyribonucleic acid and is contained in every single living cell in the entire world. Deoxyribose is ribose with one oxygen missing, which makes it structurally stronger. DNA acts as the blueprint for our cells. Think of your cell as a construction site, whereas the proteins are the brick and mortar of the buildings, while the construction workers are the membrane-bound organelles. So what tells the workers what to do? DNA. DNA is the construction site manager, who contains the blueprints of all processes that occur in the cell.
What actually makes up DNA? It is made up of two chains of building blocks called nucleotides, which are composed of phosphate groups, deoxyribose groups, and a nitrogen base. It's held together by a hydrogen bond. DNA is a double helix, which actually resembles a twisted ladder. If we untwist the molecule, we can take a better look at its structure. The side rails of the ladder are constructed of the phosphate and deoxyribose sugar portions of the nucleotides. The irregular black piece represents a deoxyribose group. The white tube represents the phosphate group, or the side rails of the ladder. The rungs of the ladder are made up of 4 nitrogen bases. The blue tube represents the cytosine nitrogen base. Cytosine is a single-ring nitrogen base called a pyrimidine. The green tube represents the thymine nitrogen base. Thymine is also a pyrimidine. The orange tube represents the adenine nitrogen. Adenine is a double-ring nitrogen base called a purine. The second purine, guanine, is represented by the yellow tubes and is also a double-ring nitrogen base. The hydrogen bond that holds the nucleotides together is a small white rod. Notice how the white tube and the white rod look similar. However, the white tube is longer in length and hollow in the center, so that you can tell them apart when we build our DNA model. Now let's build our DNA. First, we'll begin by connecting our black piece with the white hollow tube to make our side rails, which symbolize the deoxyribose sugar with the phosphate group. Next, we need to attach our nucleotides and connect them with the hydrogen bond, represented by our small white rod. The green, blue, orange, and yellow tubes may be attached in any random order. Notice how the nitrogen bases on each side of the rail don't match. This is due to the base pairing rule. The base pairing rule states that thymine bases are only paired with adenine bases, and guanine bases are only paired with cytosine bases. In other words, green tubes only pair with orange tubes, and blue tubes only pair with yellow tubes. The order in which the nitrogen bases occur in our individual DNA ladders is what makes up the unique blueprint or genetic code of a living thing. In fact, every organism on Earth has a different number and order of base pairs, which is what makes all living things unique and diverse. Now that the DNA model is complete, we can twist it to go from a ladder to a double helix. The double helix structure of DNA is essential because it allows a twisted ladder to wind and unwind so that the nucleic acid chain can duplicate itself. This process is known as replication.
Replication happens every time a cell divides. While DNA structure resembles a twisted ladder, replication can be compared to a zipper by untwisting and flattening the model, and then beginning to break each nitrogen base pair apart. This causes the zipper to begin to fork and take on more of a Y shape. The side rails of the zipper are still constructed of the phosphate and deoxyribose sugar proteins of the nucleotides, just like the DNA structure. But when DNA double helix continues to unzip, new nucleotides consisting of the deoxyribose sugar, phosphate, and nitrogen base will then pair with the original nucleotides. The new nucleotides still follow the base pairing rule, and the hydrogen bond, or the small white tube, is still added to connect the two bases. At the end of the double helix, the two strands of DNA have completely separated and we have two identical strands. They are not complete yet though. Adding new side rails is necessary to complete our ladder. Once the deoxyribose sugars and phosphate group are connected, we have officially manufactured two identical strands of DNA to replace the sole original strand.
Deoxyribonucleic acid is a blueprint of our cells. Because of this, each of us has a unique identity stemming from the different number, order, and pairing of the nitrogen bases within our DNA. DNA structure appears as a ladder, which allows us to easily grasp the concept of the base pairing rule and how it replicates itself.